Thanks, Stuart and Mark and Baker McKenzie for hosting us. And hopefully in the next 20 minutes, I'll explain to you why we are very excited about the molecule Xanamem that we are developing. And we really believe that it is a new treatment for depression with a novel and safe mechanism, and similarly can significantly halt the progress of Alzheimer's and potentially other neurodegenerative diseases. So do I click?
Just press down with the down button there. Or you can click to it.
A down button.
Oh, goodness.
The down button?
Russian interference there.
That button.
How we do that one?
Very good. All right. I will make some forward-looking statements, but I won't make you read that. So before we get into the details of some of the presentation, I'll just sort of summarize the situation for the company. The molecule has a novel mechanism, and that mechanism is to control brain cortisol. So for many decades, in fact, in the case of depression, more than 7 decades of research have associated elevated levels of cortisol, sometimes in the blood, which is sort of difficult to measure because of variability, but in brain fluid as well, with depression. But nobody's ever had a tool molecule that could actually get into the brain, control tissue cortisol levels in the brain without interfering with your adrenal glands' normal synthesis and control of your normal stress mechanisms. And essentially that's what our drug Xanamem does. It's a once-a-day oral therapy. It has extremely good pharmaceutical properties, so it's only a low dose of 10 milligrams. It's got nearly 100% bioavailability and virtually no drug interaction potential with other small molecules or large molecules. So at this stage, we've treated nearly 400 people. It's got a great safety profile. So from a development point of view, it's a really promising looking molecule. We're going after large markets and we're not at this stage, we don't believe we need to subset any of these markets. It looks like our drug is clinically active in most, if not all, patients with depression. And similarly for Alzheimer's disease, we're going for a fairly broad patient population. We've got good composition of matter protection to 2031 with automatic 5-year extensions in virtually all markets. I think India might be the exception. So that gives us good protection to 2036. And we have a number of patent families that have been generated for manufacturing use, for example, for depression, that gives us protection probably well into the 2040s. So many of you will have seen that we announced a capital raising. Doing these phase 2A and then now phase 2B trials is not necessarily cheap, but we do run a very lean ship doing our own clinical operations to keep clinical trials costs down, and of course benefit from the R&D tax credit, which we expect this year, for example, to be $9 million. So it's quite a big source of additional non-dilutive funding for shareholders. So I myself have put in an extra $1 million into this round. We're doing an 8.1 million placement to selected new institutional investors. Pleased to see some really good investors coming onto the register. I'm happy to put that second millions. It's roughly 2 million of my own money into this company. And I don't do this capriciously. The reason I took this job 3.5 years ago when I came back from the US was that after 3 months of diligence, I actually did decide that this was a de-risked Alzheimer's disease molecule, which sounds like an oxymoron, but I'm pleased to say that different clinical trials we've done and the data we've generated seem to be proving that I was right. And I do like to be right. So hopefully I'll continue to be right. So what does this new funding do for us? It totally takes this issue of we need to raise money for our trials off the table. We have the new Alzheimer's phase 2B study fully funded, and we will get interim data from that study in the middle of next year and the final results the year after. So I'll go into explain a little bit more in the next few slides. So we do have an experienced team at the board level and also the management team. A number of the management team are based in the US and also one of our board members, Nikki Vasquez. So we essentially are a Sydney-based HQ, but we have full-time employees in almost every city in Australia and operate in a virtual model, and about a third of our team are based in the US, some of them experienced manufacturing people who worked with me at Principia Biopharma, the company that Stuart mentioned before. So we're about 20 people, probably all up, and we don't anticipate needing to grow. We think we can supervise the clinical trial program, manufacturing, and regulatory interactions that we need with that level of a team. So quick stock market chart, lots of ups and downs. I think we were valued at 300 million at the top of the frothy 2021 peak soon after I took the job. Thought it was all about me, but it wasn't. It was all about the market. But look, the fundamentals of the company are very strong. We're roughly currently 100 million market cap. I think there are many companies with less good clinical data in depression, for example, or in Alzheimer's, valued at many multiples of that. Recently, we had a run-up leading up to our phase 2A depression trial. The top line for the cognition endpoint was negative, but I'll show you very positive depression data from that trial that establishes the molecule and validates the cortisol mechanism as something that's really highly clinically relevant. So I'm the second biggest shareholder. Biotech Value Fund in the US is the largest shareholder, and we have a very dedicated and sticky top 20 shareholder register, but we are largely retail, which in Australia means that you need to talk to the various investors who expect you to have timelines similar to mining rather than pharmaceutical development. So that is an ongoing issue. And I won't dwell on this, but neuroscience seems to be the new cancer. People are really excited as new biomarkers are coming into the field. And hopefully what's going to happen is that Alzheimer's disease, for example, will turn out to be multiple different diseases where patients get matched to the right combination of new therapies, much like cancer. Breast cancer was breast cancer 30, 40 years ago. Now it's many different diseases with many different treatments. So we do believe that neuroscience is going the same way. So Lou Gehrig's disease, ALS, Alzheimer's, I think we're not there yet, but that's where the field is heading. And Big Pharma is starting to invest in new psychiatric drugs, such as these big deals for Karuna and Cerevel of $9 and $14 billion last year, but also investing in Alzheimer's disease efforts as well. So I'm going to talk first a little bit about our Alzheimer's program, and then I'll show you the results from our very recent phase 2A data in depression. So the recently released depression data give us very good confidence now that our dose is right at 10 milligrams and that controlling brain cortisol really does work. And so that means that we have increased confidence in this phase 2B/3 study that we are doing currently for Alzheimer's disease. It's enrolling in Australia. US sites will start enrolling next week, and we anticipate being able to accelerate that program. At the same time, we plan to strengthen the design a little bit and to try and ensure that we can get this to be one of the 2 pivotal trials the FDA would accept for approval. So we're now really flipping from the phase 2A, phase 1 pilot early stage clinical trial program for depression and for Alzheimer's disease into 2B/3 trials where we're really focusing on what are the regulators going to ask for to get the drug approved. And so we are anticipating having a generously sized safety database of at least 1,500 patients, 100 of which will be treated for more than 12 months. And as well, we're using an endpoint in this trial called the CDR sum of boxes, was used by Eisai for their recent antibody approval. We're having an independent data monitoring committee to make sure everything statistically is kosher, and as well the statistical methods for the trial. Look at the endpoints in sequence so that you can actually claim less than 0.05 for multiple endpoints potentially. We've also got a commercial formulation of our tablet, which is great. So that's being used as well. That's also a necessary thing for a pivotal trial. And the slide just to talk about the market, but I think no one needs to argue about the size of the Alzheimer's market potentially. But essentially, there are other companies in the world looking to develop a pill for Alzheimer's. A lot of those have an amyloid-related mechanism. The amyloid protein that is the hallmark of Alzheimer's disease it's pretty much been shown by the work with these amyloid antibodies to not be the cause of Alzheimer's disease. So really, we think our molecule Xanamem controlling cortisol has a really good shot at being the first safe and effective oral therapy for Alzheimer's, and as such would leapfrog to first-in-line therapy. So our analyst estimates peak sales at $5 billion in the 2030s. I think it could easily be more than that. So how does it work? So we've been struggling with how do you explain the pharmacology and biology of cortisol to people who are not deeply educated in the pharmaceutical sciences. And probably the easiest way is to say that stress becomes chill. So we all need our stress response. We all need our cortisol, so that if somebody invades your house and attacks you with a knife, you get adrenaline pumped out. You also get cortisol pumped out by your adrenal glands. And that helps your muscles and your brain, helps you, fight the intruder or run away. However, that chronic level of stress, which is associated with depression and Alzheimer's disease, is bad for you. And you heard from one of the previous speakers that chronic therapeutic steroids are very bad for you, lots and lots of side effects. Well, it appears to be true when you have your steroids coming naturally from tissue production in the brain and elsewhere in the body. So, and cortisol works in a couple of different ways. One is a fast mode, changing things without changing protein synthesis. And there are slow changes, which are genomically mediated, which actually change the pattern of receptors and proteins produced in cells. So our drug has potential to essentially bring the brain back to that chill state, which is hopefully something that would not just reduce the chances and treat Alzheimer's disease, but it possibly may be related to programmed aging and improving longevity as well. Now, we're not developing the drug for that, but it is an interesting concept. And one of the things that I was thinking about as I heard one of the earlier presentations was it's really important to note that you've got the right dose. And this is a human PET study that was done actually before I joined the company. One of the main reasons that I took the job. And on the left, you see the bright green and gold, the distribution of this 11-beta-1 tissue cortisol production enzyme in the brain. And on the right, after just 7 days of daily therapy with Xanamem, you see near total abrogation of that signal, meaning the drug is sitting on that enzyme blocking the tracer binding. And you only need 30 to 60% inhibition of the target to have full abrogation of cognitive decline in an animal model. So we know that this dose we're using of 10 milligrams in the center there is very much a safe and effective dose and should have a full clinical effect. So what of the antibodies that have been recently approved? So there was much hype a couple of years ago when the FDA only approved one antibody called aducanumab on one positive phase 3 trial, and the other trials were negative. It was very controversial. And the reason it's controversial is that they don't work very well. And even the ones that have been subsequently approved by from Eli Lilly and from Eisai, essentially they all follow in an 18-month trajectory, what you're seeing here on the screen. The placebo decrement in function in these Alzheimer's patients is shown in the dotted line. And when you treat with an antibody, you move that progression, you slow it a little bit, and that's the solid blue line. But where you really want to be, so if I get memory impairment and get diagnosed with early Alzheimer's disease, I want to take a treatment that puts me on the horizontal top line there, where I don't progress any further. And in fact, so that Alzheimer's disease would become a treatable disease, much the way HIV has and certain types of cancer. And that is where we're heading, but we are a little bit handicapped because the true causal biology of Alzheimer's is not really, really known. But if you just compare this to what we have in our own data, we went back and looked at our phase 2A trial that was done without modern biomarkers and went back, fortunately had people who could reconsent to having new biomarkers done in a new double-blind in silico trial. And what we found in patients with elevated baseline p-tau, which is a very important biomarker that means that you've got amyloid protein in your brain and probably you're more likely to progress. You can see here in the purple line, the much slower progression in the Xanamem treated group versus placebo in the grey. And this was nearly statistically significant despite there only being 34 patients in this sample. But as a pilot data set to help us design the next clinical trial, it's a fantastic piece of data. And so this is what we've done. We're essentially running this trial currently. It's a larger placebo-controlled trial of 10 versus placebo over 36 weeks. we are taking people who've got mild to moderate Alzheimer's, elevated blood p-tau, and then the primary endpoint will be the same endpoint I showed you on the previous slide, this global assessment score called the CDR sum of boxes. We'll measure lots of other things as well. And currently we're enrolling here at 15 Australian sites, and we'll probably have at least 10 US sites in the very near future, with results mid next year and the year after. So very excited about the prospects based on a big weight of evidence, not just now that cortisol is associated with Alzheimer's, but we have now shown that intervening with our drug has an effect to improve depression and in this biomarker analysis, slow progression of Alzheimer's disease as well. So now we just need to prove it in a pivotal phase 2b/3 trial. So just want to speak briefly about the depression data. This was a very different trial. So this is a phase 2a, essentially a learning proof of concept trial, if you like. This was 10 milligrams again, the same dose of Xanamem, on top of an existing antidepressant in more than 80% of cases. So these are people who are heavily pretreated. They still have depression despite being on antidepressant therapy, but they're not the super treatment resistant population, such as might take ketamine or one of the new or electroconvulsive therapy, for example. So they're people who are pretty severe, but they are not yet in that super treatment resistant group. And they had to have cognitive disability to half a standard deviation below on a coding test to get into the trial, because we were interested to see, based on our healthy volunteer work, whether our drug could improve cognition in these patients, because foggy thinking is a key feature of depression. And the key secondary endpoint was depression itself, measured in a couple of different ways. And just to explain one thing here, I won't go through this all, but we do use in these early trials the concept of an effect size, and that's called a Cohen's d statistic. It's essentially a proportion of the standard deviation or variability at baseline. So if you imagine people have an average of 2, the standard deviation might be 1. So we're hoping to see a large clinical effect, which would be an improvement of half a standard deviation between placebo and the active therapy. So you'll see this Cohen's d statistic in a moment. So the patient population was very much what we expected, average age of around 50, more than 80% were on antidepressant therapy, and the baseline depression scores indicated moderately severe depression. And the top line results are shown here. We did see clinically and statistically significant treatment benefits on depressive symptoms and measured or displayed in 3 different ways, which I'll show you. Interestingly, cognition improved in both the Xanamem and the placebo group in a way that we weren't able to see a greater benefit in the Xanamem versus the placebo. And this is something, placebo effects in these trials are famous and common. So the trial was well conducted. And I realize my timekeeper is here. These are the data for depression. And on the left, you see the separation of the curves for Xanamem in purple and placebo in gray at week 6, and then statistically and clinically significant, interestingly, 4 weeks after stopping therapy. And on your right, you see the patient-reported outcome, where the curves separate much earlier and remain separate as well at 10 weeks, at week 10, meaning 4 weeks after therapy. So, lastly, I'll just show you the responder analysis. 50% more responders in the Xanamem group for both a major reduction in this MADRS depression score or complete remission in depression, which is getting to a score of less than 10 points. So good subgroups, good safety, and a large market also for a new antidepressant with good safety and good efficacy data. So our conclusions are controlling cortisol in the brain really works. After many decades of research, no one else has had a molecule that actually does what it needs to do to prove this cortisol hypothesis, if you like. And this sets us up now for key pivotal trials.